Rosalind Franklin was a brilliant scientist who made a crucial discovery that changed the course of biology. She was born in London in 1920 to a wealthy and influential Jewish family. She had a passion for learning and science from a young age, and excelled in her studies at St. Paul's Girls' School and Newnham College, Cambridge. She graduated with a degree in natural sciences in 1941, and then pursued a PhD in physical chemistry under Ronald Norrish at Cambridge. However, she soon became frustrated with his lack of guidance and interest in her research, and decided to join the British Coal Utilisation Research Association, PURA, in 1942. There, she studied the structure and properties of coal, and made important contributions to the field of carbon science. She earned her PhD from Cambridge in 1945, and published several papers on her coal research. In 1947, Franklin moved to Paris, where she joined the Laboratoire Central de Services Chimiques de l'État, led by Jacques Mering. There, she learned the technique of X-ray crystallography, which uses X-rays to reveal the atomic structure of crystals. She became an expert in this method, and applied it to study various substances, such as DNA, RNA, and viruses. She also developed a close friendship with Mering, who was a mentor and a colleague to her. In 1951, Franklin returned to London, where she was offered a research position at King's College, under the direction of John Randall. She was assigned to work on the structure of DNA, along with Maurice Wilkins, who was already working on the same project. However, Franklin and Wilkins had a tense and strained relationship, due to their different personalities, approaches, and expectations. Franklin insisted on working independently, and did not appreciate Wilkins' interference or suggestions. Wilkins, on the other hand, felt that Franklin was intruding on his territory, and did not respect her authority or expertise. They also had conflicting views on the nature of DNA, with Franklin favoring a helical structure, and Wilkins favoring a paracrystalline one. Despite the difficulties, Franklin made remarkable progress in her DNA research. She used a new type of X-ray camera, designed by Raymond Gosling, a PhD student who worked under her supervision. She also improved the quality of the DNA samples by using a humidifier to keep them hydrated. She took hundreds of X-ray photographs of DNA and analyzed them with mathematical calculations and measurements. She discovered that DNA had two forms, A and B, depending on the humidity level. She also found that DNA had a regular and repeating structure, and that the phosphate groups were on the outside of the molecule. She deduced that DNA was most likely a helix, with more than one strand. One of the most famous photographs that Franklin took was photo 51, which showed a clear and sharp image of the B form of DNA. This photo was taken by Gosling in May 1952, under Franklin's direction. It revealed the characteristic X-shaped pattern of a helical structure, and provided crucial clues to the dimensions and symmetry of DNA. However, Franklin did not realize the full significance of this photo at the time, and did not publish it immediately. She continued to work on refining her data and calculations, and planned to present her results at a conference in Naples in 1953. Meanwhile, Wilkins, who had access to Franklin's data and photos, showed photo 51 to James Watson, a young American biologist who was visiting King's College in January 1953. Watson was working with Francis Crick, a British physicist, at the Cavendish Laboratory in Cambridge on building a model of DNA. They had been inspired by the work of Linus Pauling, an American chemist who had proposed a helical structure for proteins. When Watson saw photo 51, he was amazed by its clarity and beauty, and realized that it confirmed his and Crick's hypothesis of a double helix. He rushed back to Cambridge and told Crick about the photo. They then used the information from the photo, along with the data from Franklin's and Wilkins' papers, to construct their famous double helix model of DNA. They also incorporated the idea of complementary base pairing, which was suggested by Erwin Chargaff, an Austrian biochemist who had found that the amounts of adenine and thymine, and of guanine and cytosine, were equal in DNA. They completed their model in February 1953, and published their paper in Nature in April 1953, along with 2 other papers by Franklin and Wilkins, and by Gosling and Franklin. Franklin was not aware of Watson and Crick's work until after they had published their paper. She was not consulted or credited by them, and she did not receive a copy of their paper until after it was printed. She was surprised and annoyed by their model, and felt that they had used her data without her permission or acknowledgement. She also found some errors and inconsistencies in their model, and questioned their assumptions and evidence. However, she also recognized the elegance and simplicity of their model, and admitted that it was likely to be correct. She decided to continue her own research, and to verify or refute their model with more experimental data. In 1953, Franklin left King's College, and joined Birkbeck College, where she worked under John Desmond Bernal, a renowned crystallographer and physicist. She switched her focus from DNA to viruses, and started to study the structure of tobacco mosaic virus, TMV, a plant virus that causes a mosaic pattern on the leaves of infected plants. She applied her X-ray crystallography skills to TMV, and made several important discoveries. She showed that TMV had a helical structure, similar to DNA, and that it consisted of a single strand of RNA, surrounded by a protein coat. She also determined the dimensions and arrangement of the RNA and protein units, and proposed a model of how they interacted. She published her findings in Nature in 1955, and received international recognition and acclaim for her work. She also collaborated with other scientists, such as Aaron Klug and Donald Casper, to study other viruses, such as polio virus and turnip yellow mosaic virus. She made significant contributions to the field of structural virology, and laid the foundation for understanding the molecular basis of viral infection and replication. Franklin had a short but productive career, and published more than 50 papers in her lifetime. She was respected and admired by her peers and colleagues, and was nominated for several prestigious awards and honors. She was also offered several positions and fellowships at various institutions, such as the Royal Society, the University of Cambridge, and the University of California, Berkeley. She had plans to visit the United States, and to continue her research on viruses and DNA. However, her life and career were cut short by a tragic illness. In 1956, she was diagnosed with ovarian cancer, which was likely caused by her exposure to X-rays. She underwent several surgeries and treatments, but they were not effective. She continued to work and publish papers until the end of her life, despite the pain and suffering. She died on April 16, 1958, at the age of 37, in London. She was buried in the Willesden Jewish Cemetery, next to her father. Rosalind Franklin was a remarkable woman who made a lasting impact on science and society. She was a pioneer in the fields of biophysics and molecular biology, and a role model for women in science. She was a dedicated and meticulous researcher who pursued the truth with rigor and integrity. She was also a generous and compassionate person who cared for her family and friends, and supported various causes and charities. She had a curious and adventurous spirit, and enjoyed traveling, hiking, skiing, and photography. She had a love for life and learning, and a passion for science and discovery. She was a hero who deserved more recognition and appreciation for her work and achievements. She was Rosalind Franklin, the woman who revealed the secret of life.